Hello, welcome to We Speak. My name is Rich Roven, and I'm a neurosurgeon and medical director of the Upper Michigan Brain Tumor Center. For those of you new to We Speak is an online journal club where authors present and discuss one of the recent research publications. With We Speak, we hope to make biomedical research more understandable, transparent, and collaborative. Tonight we're very pleased to welcome Keith Sabin as our guest lecturer. Keith will have a more formal introduction in just a few moments. Tonight's moderator is John Lawrence with the Department of Biology here at Northern Michigan University, and John was the first postdoctoral fellow at the Upper Michigan Brain Tumor Center. Our panelists tonight are Robert Wynn, John Lawrence, and myself. We encourage you to ask questions. You can tweet a question at us with the hashtag WeSpeak. We are monitoring Twitter for your questions. You can also visit our Google Plus page and use the live Q&A feature. And you can go to the Upper Michigan Brain Tumor Center YouTube page and watch there as well. And with that, I'm going to turn the presentation over to John Lawrence, and we'll talk to you soon.
Good afternoon. I'm John Lawrence, and I'm an assistant professor here in biology at Northern Michigan University. I was the first postdoctoral fellow of the Upper Michigan Brain Tumor Center, as Dr. Roven mentioned. Things that I investigate here at the lab are the roles that hormones and nervous system play in brain tumorigenesis. I'm interested in environmental enrichment, neuroplasticity, exercise, and other alternative types of medicine on the quality of life in brain tumor patients. And with that, I'd like to introduce Dr. Robert Wynn, who is the Director of Research for the Upper Michigan Brain Tumor Center. And you can have the floor, Rob.
Thank you, John. As Dr. Lawrence mentioned, I'm Dr. Rob Wynn. I'm a PhD and a professor in biology at Northern Michigan University, as well as the research director for the Upper Michigan Brain Tumor Center. And I had the great privilege of being Keith Sabin's master's advisor when he worked on his master's degree at Northern Michigan University. Keith, as Dr. Roven indicated, did his undergraduate and his master's education at Northern Michigan University. A couple of things I can tell you about Keith. One, I don't know that I've ever met anyone who is more excited or wanted to be a scientist more than Keith Sabin. When we found an interest in science, he came to my office often, asking if he could become involved, and once we allowed him that opportunity, I don't know that anyone's ever taken the bull by the horns like Keith did. Really ran with that opportunity, and I know that someday Keith's name is going to be well known throughout the scientific community. And so with that, it's my pleasure to introduce Keith Sabin, who's going to talk to us about microvesicles and their role in regeneration. So I'm pleased to turn the time now over to Keith.
All right, thank you very much for that kind introduction. So without further ado, I'll go ahead and get the show on the road. Hopefully, bring my slides up here. There we go. So like Dr. Wynn had mentioned, I will talk to you guys today a little bit about tissue regeneration, stem cells, and microvesicles. and I am currently a PhD student in the Molecular, Cellular, and Structural Biology program at the University of Minnesota in the Twin Cities. So just to give you a little bit of an overview for what we'll be talking about today, I'll start off by giving you an introduction into what is regeneration. And to do that, I will walk you through a couple of the different model organisms that are widely used in the field of regenerative biology. And from there, we'll move on to how mammals respond to injury and their wound healing response. And then after talking about that, we'll go through and I'll give you an introduction to the newly emerging field of regenerative medicine, and specifically how stem cells and stem cell transplantation kind of fits into that new medical field. And from that, that'll segue into the field of microvesicles, which is what I focused on primarily when I was at Northern and the Upper Michigan Brain Tumor Center for my master's. And then I'll kind of end the talk with a future directions type of pitch to you. So just to kind of start out here, so what is regenerative biology? So this is where we study the ability of certain organisms to regenerate following injury. So some model organisms that are widely used include Xenopus laevis, which is the African clawed frog. And I'll have pictures of these critters a little bit later on. But they're able to regenerate their tail and limbs as well as their lens following injury. So if you cut off their leg or their tail, they can grow that back during their tadpole stage. However, once they metamorphosize into adult frogs, they lose that potential completely. So that's kind of an interesting model where we can study the life stage of this animal when it can regenerate and also when it cannot regenerate. So that's kind of useful for comparative studies. Another organism that's widely used is the zebrafish. So the zebrafish is able to regenerate its fin and heart and spinal cord throughout its entire lifetime. So one interesting thing, though, about spinal cord regeneration in zebrafish is while motor neurons regenerate, sensory neurons do not. And then the model organism that I'm working on currently in my lab, or in the lab that I work in, is the axolotl, which we kind of consider the champion of regeneration. So it's a salamander, and axolotls, so this is a full picture, kind of cute. Axolotls can regenerate their jaw, they can regenerate lens tissue, they can grow back limbs after amputation, as well as following kind of ischemic heart injury. They can regenerate that tissue as well as tail tissue. So you can see there's a diverse tissue types that the salamander is able to regenerate. And so this figure here, I think, illustrates very nicely the ability of certain organisms to regenerate at certain life stages and other organisms to not. So down here, kind of on the x-axis, we have their ability to regenerate. So you have complete regeneration following injury, or wound healing with scar. So scar formation is usually opposed to functional regeneration. So in a mouse model, which would be most translational to humans, in their kind of early embryo to neonatal stage, they do exhibit an ability to regenerate their skin as well as fingertips or paw tips, but however they lose that ability when they become adults, and then they actually get scar formation. And like I alluded to earlier, Xenopus, or the frog model, can have complete regeneration at the tadpole stage, but again, in the later stages of its life, it is unable to regenerate functionally. Whereas Newton salamanders, throughout their entire lifetime, they can have complete regeneration. So I think this figure illustrates it very nicely. So this is an amputation, a limb amputation in a newt. And as you can see, when you remove part of the limb, after about a week, you get a formation of this structure, which is called the blastema. So this little bud here is what gives rise to all of the regenerating tissues for your newly formed limb, which is going to functionally and morphologically look like a limb after about 42 to 70 days following amputation. And one of the really interesting things about this model is, so in these panels here, right here, you amputate the arm more distal, so closer to the hand, whereas over here, right, you are going to amputate more close to the shoulder. So regardless of where you amputate, the new limb that's developing actually knows how much needs to be regenerated. So you get regeneration of the full arm and digits in this more proximal amputation, whereas you only get regeneration of these more distal structures, so your finger structures, which I think is a really interesting phenomenon in amphibian regeneration. So for these different model organisms, so here we have the axolotl, this is Xenopus laevis, and the zebrafish. These are really good model organisms for studying what I would refer to as organ regeneration, so like tail and limb amputations. So these are going to be complex tissues that are going to include cartilage, connective tissues, bones, blood vessels, muscle, epidermis, etc. But then to study more kind of tissue regeneration, so following injury, if you have your kidney cells start dying, or if you have a myocardial infarction or a heart attack, to study heart regeneration in a more translational sense, the mouse is kind of the predominant model organism that people use. So then, in this particular example that I'm giving here for wound healing, this is specifically looking at injury in the skin, but certain general concepts are conserved in mammalian wound healing regardless of the tissue type. So you have your initial wound, so if this is a scratch on your skin, or if it's ischemic injury in your heart, so if you've had a heart attack, you have initial damage to these cells. The cells will die, and you will get formation of a blood clot. And then later on, during the wound healing process, you kind of get thickening of the epidermis here. But really the major thing that's detrimental to regeneration in mammals is we tend to produce a lot of collagen, or extracellular matrix components. So that's kind of the scaffold then that the cells of your tissue are going to use to organize themselves. So in mammals, we produce excessive amounts of collagen, which is actually inhibitory to functional regeneration. Whereas in axolotls, so this is a staining of the axolotl skin, so you have the epidermis here on top with the dermis just underneath it. So you do get thickening of the epidermis like you do in mammalian systems, and you still get extracellular matrix or collagen synthesis. However, something that axolotls can do that mammals don't is after you get this buildup of collagen, they will remodel the tissue and get rid of unnecessary collagen components. And then that's going to allow for overall a functional regeneration. So again, like I said before, this specific example is from the skin, but this kind of holds true in spinal cord injury or limb amputation, heart regeneration, etc. As mammals, we have this buildup of collagen and extracellular matrix that's detrimental. So now moving on, we'll talk a little bit about, like I said before, kind of the newly emerging field of regenerative medicine and how stem cell transplantation kind of fits into that field. So within the last decade or so, I think the idea of regenerative medicine, as we start to understand more about the basic biology of stem cells and their potential therapeutic uses, has really exploded in this field. And really, the main focus of this discipline is to stimulate tissue regeneration following injury or in the context of degenerative diseases. So if you lose an appendage, or if you have a heart attack, or are in stage 4 kidney failure, regenerative medicine is really looking at how can we activate the body's repair mechanisms to functionally repair these tissues so that your heart contracts with the same amount of force, or your kidneys can start filtering the blood again. So there are many different ways that we can address this question. I'm going to focus specifically on cellular therapy, so this will be in the context of using stem cells to repair damaged tissue. However, towards the end of the discussion, I will touch a little bit on gene therapy and how that's being applied in a regenerative context. But just be aware that there are other avenues that you can take to address this concern. So just to, I guess, acclimate everybody with the stem cell hierarchy and their ability to differentiate or turn into other cells. So right up at the tippy top of the hierarchy, we have totipotent stem cells. So these are going to be cells that can turn into any of the extra embryonic tissues. So this is going to be structures such as the placenta, as well as any of your somatic cells, which are any of the cells in your tissues, or your germ cells, which are your sex cells, so your sperm cells or your oocytes. And then from there, through development, you get pluripotent stem cells, which are analogous to this blue blob here. So that's called the inner cell mass, and this is what embryonic stem cells are derived from that we use in culture systems in the lab. And these cells are able to form somatic cells or your sex cells, but are no longer able to form the extra embryonic tissue or your placenta. And then through that, we have multipotent stem cells, and these are going to be lineage restricted. So we have stem cells in our gut, in our intestines, that can give rise to new gut cells. And we also have adult stem cells, I guess, in your brain, which give rise to neural cells, so neurons, ependymal cells, different glial cells, etc. And then from there, you finally have, I guess, the least potent cells are going to be your terminally differentiated cells, and those are going to be muscle cells or skin cells or fat cells. So in the context of what I'll talk to you today about, we're going to focus primarily on these multipotent cells or adult stem cells, if you want to think about them that way, because these are stem cells that persist in adult tissues, is why they're called that. And specifically, there are these things called mesenchymal stem cells that reside in different niches or different areas of your body, and these are the most widely studied stem cell in the field of regenerative medicine. And they're very easily isolated from different sources, so bone marrow or fat cells. And one really nice thing, one really clinically nice thing about these cells is they can be isolated from a patient. Those patient cells can be grown up in the lab, and then those cells then can be injected back into that patient. So this is extremely advantageous in that it is the patient's own cells that we are using. We're not introducing any foreign material into this individual. So we would have to worry about immune rejection and different complications that would arise through that process a lot less, since it is the patient's own cells. And one characteristic of these cells that make them very appealing to the field of regenerative medicine is that when you inject mesenchymal stem cells into an injured individual, these cells will actually home, or they will specifically target these injured tissues, and they will reside there and mediate tissue regeneration or tissue repair in the site of injury. So currently there are 340 clinical trials using mesenchymal stem cells to treat tissue injury and degenerative diseases that are registered with the National Institute of Health. So this is, technology that is being developed and being applied in the clinic. And I think it'll be really interesting to see in the next, decade or 20 years or so where this takes us. So in the field of regenerative medicine, in the idea of stem cell transplantation, at least as far as mesenchymal stem cell goes, there's really two prevailing hypotheses as to how they are conveying these beneficial traits. So there's the engraftment theory, which suggests that mesenchymal stem cells are injected into an injured mouse, and they home into the injured tissue. And once the stem cell gets to the site of injury, it will then actually engraft, or it will place itself into the injured tissue, where it will divide and differentiate into that cell type. So if you have kidney failure, these mesenchymal stem cells will home to your kidney, it will engraft into your kidney, and then it will divide and turn into kidney cells. And in that way, it will repair the damage. So it will directly engraft and then repopulate an injured tissue. And there is some evidence for this. For a long time, this was thought to be the prevailing mode or mechanism for mesenchymal stem cells. So in order to show this, what they've done here is, so on A, what they've done is they've labeled the Y chromosome in male mice. So males are XY, they have the Y chromosome, females are XX. So what they've done is in lung tissue, they've labeled the Y chromosome here in yellow, and it's really handy. They have arrows pointing exactly where they want you to look. So then what they can do then is they can isolate stem cells from this male mouse, and then they can induce lung injury into female mice, and then inject the male stem cells, and then they can look for the Y chromosome in these injured tissues, and they use that as a marker to tell them whether or not these stem cells are engrafting into the tissue. So they do claim that there is some engraftment that's occurring, and then they have certain functional metrics that they look at to say that yes, these stem cells are facilitating functional regeneration. However, recently in the literature, there's this whole idea of a paracrine theory, in that these mesenchymal stem cells are going to be injected into injured mice, and then they're going to release certain factors at the injury site and into circulation, and it's actually going to be these secreted factors that promote proliferation and inhibit cell death of these injured host cells. So that stem cells are actually contributing to tissue repair by modulating the host wound response via these secreted factors, and are not directly implanting into the wounded tissue itself. And again, so this is a similar, a different article, but a similar experiment where they, in A here, they have male mice, and again, they're probing for the Y chromosome, which is shown here in red, and this is actually kidney tissue, so this is a different model of, or from this particular paper was a different model of injury. So then in their injured female mouse here, after stem cell injection, they showed that there was no stem cell engraftment. However, there was still functional regeneration that occurred in these mice. So their argument was that this is independent of engraftment. And kind of as time unrolls, we see that there's a lot of evidence that supports this paracrine hypothesis, in that there are these secreted factors that these stem cells are releasing that's actually modulating the host response and promoting tissue repair that way. So what is it that these stem cells are secreting? So they're going to secrete factors that are going to modulate the immune response. So what immune cells come to the site of tissue injury and when plays a huge role in determining whether or not you get scar formation or functional regeneration. Similarly, they can release factors that promote blood vessel formation or angiogenesis. So if you have an injured tissue, obviously you want really good blood supply so you can get nutrients and take away waste products. It also is going, these factors are going to promote cell division and cell growth or proliferation of the host cells. And they're going to provide pro-survival factors to the injured host cells as well. So that's going to make it so those cells no longer die. They're going to promote survival of these cells. So the specific identities of some of these factors are transforming growth factor beta, or TGF-beta, hepatocyte growth factor, vascular endothelial growth factor, and epidermal growth factor. So these are all secreted molecules that these stem cells are going to release. And more recently, a role for microvesicles has been shown in mediating these pro-regenerative or pro-repair phenotypes. So I'm going to talk to you more now about what are microvesicles exactly, where do they come from, what's in them, and what do they do. And from that, I'll give you kind of a more comprehensive view on how specifically microvesicles play a role in tissue repair. So really, microvesicles is a more generic term to refer to any membrane-bound vesicle that is released by cells. And the two predominant types of vesicles that are released by cells include exosomes and microparticles, which I'll speak more about later. But exosomes come from the endocytic pathway. So in a normal cell, at any given second, what happens is the membrane of the cell is going to invaginate, and you, through the process called endocytosis, so you have these small membrane-bound vesicles that come into the cell, and they accumulate, and they form this structure that's called an early endosome. So now this is a large membrane-bound vesicle that's within the cell. So these early endosomes can be used in different metabolic pathways in the cell, or they themselves can actually accumulate smaller vesicles inside of them. and at that point they're called multivesicular bodies, right? Because it's a vesicle with with multi-smaller vesicles inside of them. So these multivesicular bodies then can actually fuse back with the cell membrane and release these smaller vesicles into circulation, into your blood supply or the local environment in a tissue, and these are what are called exosomes. So these are certain components that are enriched in exosomes. So for example, different heat shock proteins, ALEX and TSG101 are proteins that are specific to endosomes. So you would expect those to be enriched in exosomes. Ceramide is a sphingolipid, so it's a type of fat molecule that's actually enriched in these exosomes, as well as different tetraspanins and cell adhesion molecules. So then we also have microparticles, which are similar but different from exosomes. So what happens is inside of the cell, you have increased concentrations of calcium, and this increased calcium concentration is actually going to activate these different enzymes, so calpain, scramblases, flopases. And so what these enzymes do is they actually stimulate the direct budding of the plasma membrane. So you can see here you have kind of this sequential formation of these vesicles then that bud off of the plasma membrane, and they're going to be enriched in cholesterol in their lipid bilayer, as well as cell adhesion molecules, flotilin, and CD63, which is integrins and CD63 are similar components between exosomes and microparticles. So you can see how there are some unique, but definitely some overlapping markers for these distinct populations. So that's why we refer to them just generically as microvesicles, again referring to any membrane-bound vesicle that's released from cells. So why are these important, right? What's inside of these microvesicles? So it's been shown through the last decade that microvesicles contain messenger RNA, which are the molecules in cells that code for proteins. They actually have proteins in them, and interestingly, they have microRNA in them as well, which is a smaller RNA molecule which can actually influence whether or not a messenger mRNA is actually turned into protein, which is kind of a whole new level of regulation, which is pretty interesting. But then going from here, so the field of, I guess, exosomes and microvesicles has been most intensively studied in the fields of cancer biology and immunology, and is now starting to become more relevant to stem cell biology and regenerative medicine. But kind of the way that exosomes and microvesicles can function is if you have a cell type here that's going to have a cell surface protein in its membrane, right? That cell surface protein can be incorporated into the endosomal pathway, and then it can become incorporated into exosomes, and then these exosomes are going to be released from this parent cell, and it will don't. So then what happens is these cancer cells, they divide faster and they don't die as readily. So that's going to kind of confer a more, a more aggressive phenotype. and I might have been lost.
Do you need me to pull up your PowerPoint?
Oh, maybe. Is it not on the screen anymore?
No.
Oh, okay. That's unfortunate. Was I gone for long?
Half hour.
Are you for real?
No, I'm just joking.
Oh, okay. Let me see.
Hang on here.
Oh, cool. When did you lose me?
I'm having trouble calling up your PowerPoint as well. Hang on a minute.
Oh, is it not on the screen now? Okay. Let me see if, oops. Let's see here. Actually, I think I can pop it back in. Oops.
Okay.
Can you see it all right?
Yes, we can.
All right. Where'd you guys lose me?
Fast ligand.
What's that?
Fast ligand.
Fast ligand, okay.
So first panel in the immunology.
Near and dear to my heart.
Yeah, absolutely.
Okay. Right here?
Yep.
All right. So just start from the top?
Please.
Will do. Okay, so one way then that these microvesicles can function is that if you have this cell surface protein called Fas ligand that's expressed by tumor cells, it can actually become incorporated through the endosomal pathway into microvesicles, and then these microvesicles can be released into circulation where they can bind with T cells and interact with T cells, and they actually can instruct these T cells to undergo cell death. So in that way, these tumor-released exosomes can promote immune suppression, which would allow for tumor proliferation and tumor progression. An alternative method of function for these microvesicles is they can actually take membrane receptors from one cell, and then the cell releases that receptor in association with microvesicles, and that microvesicle then can transfer a functional protein to cells that wouldn't normally express that receptor. So in the context of brain tumors or glioblastoma, which is what we study at the Brain Tumor Center, cells that wouldn't normally express that receptor will start, and then that leads to increased cell division and decreased ability of these cells to be killed, which is going to confer kind of a more aggressive tumor phenotype. And then additionally, like I said before, these microvesicles can be loaded with messenger RNA and microRNA as well. So in the context of forming an immune response, when T cells are presented with antigen by antigen-presenting cells, that actually stimulates exosome release, and then those microvesicles feed back to the antigen-presenting cells, and the mRNA, which codes for protein, and microRNA feeds back to this antigen-presenting cell, and in that way, these T-cell-derived microvesicles can modulate an immune response. So while these functions, I guess, have been provided in a context-specific manner, just keep in mind that the way that microvesicles function is they can either activate cell signaling in distant cell types, they can actually transfer receptors to other cells, and they can also transfer messenger RNA and microRNA, and in this way they can influence the physiology of these cells. So I think I'm going to speak mostly to you about acute kidney injury in mice, so just kind of to familiarize yourself with this organ. So here's a picture of your kidney, and here in this close-up we can see there are these squiggly structures which are referred to as nephrons. So here's kind of a larger picture of a nephron. So nephrons are made up of renal tubular epithelial cells, and nephron is actually what will filter the blood and allow for urine production in the kidneys. So in different experimental models for kidney failure, mice can be injected with cisplatin, glycerol, or they can undergo ischemia injury, which will very nicely recapitulate human kidney failure. So in response to these different experimental models, these renal tubular epithelial cells, which make up the nephron, will undergo cell death, and ultimately results with the kidneys losing their functionality, so they're no longer able to filter blood. So interestingly, microvesicles that are released from mesenchymal stem cells have been shown to contain up to 239 unique species of messenger RNAs that can be transferred to these renal tubular epithelial cells, and these messenger RNAs encode for proteins that are involved with transcription, that are involved with translating new proteins. They're going to promote cell division and inhibit cell death, and actually promote angiogenesis or new blood vessel formation as well. And in more directed studies, so in vitro studies, researchers have induced injury to renal tubular epithelial cells and then treated them with MSC-MVs, and what they found is that you actually increase expression of BCL-XL, BCL2, and BRCA8, which are proteins that can inhibit cell death. So these are being upregulated in the injured renal tubular cells, and MSC-MVs are going to decrease the expression of caspase-1, caspase-8, and LTA, and all of these gene products are going to activate cell death. So by treating these injured kidney cells with microvesicles, you're increasing their ability to live while actively decreasing the cell death pathway, which is really, I think, pretty cool. And simultaneously, on top of doing that, so not only is it inhibiting apoptosis, it's also activating cell proliferation pathways, so ERK1 and 2, which are a major proliferation pathway. So all these things are happening simultaneously in these cells in response to treatment with MSC-MVs, which is really, I think, quite neat. And so all of these molecular events kind of culminate in facilitating functional regeneration of the kidney. So after you induce kidney injury through cisplatin or something and then treat mice with these microvesicles, they actually can functionally regenerate their kidneys, so they get back the ability to filter blood as compared to their controls, which I think is astonishing. So just kind of for the sake of time, if you are more interested in microvesicles and tissue regeneration, there are some pretty good reviews looking at cardiovascular diseases, just kind of a general stem cell-derived microvesicles and the paracrine action. So this individual, Giovanni Camusi, kind of wrote the book on MSC-MVs and acute kidney injury. So his lab is very active looking at renal failure and microvesicles, and kind of a more, not necessarily comprehensive, but in this review here that I came out with this summer, we look at cardiac regeneration, we look at kidney, liver, neural regeneration, so it's maybe more of an all-encompassing or umbrella type review. So now I want to kind of leave you guys with perspectives and give you the state of the field and maybe some future directions, which could be pretty interesting. So if you guys remember this kind of the stem cell hierarchy, I wanted to talk a moment about these iPS cells. So this is actually a really cool phenomenon that was reported by Takahashi and Yamanaka, I think in 2006. But what they were able to do was to take these unipotent, right, or terminally differentiated cells, so like your skin cells, let's say, because that's what they did, and they turned them back into stem cells. And this study literally overturned centuries of biological dogma and was one of the things that really got me interested in stem cells and regenerative medicine in the first place. But kind of how they did this is so you can take these skin cells or fibroblasts, you can introduce 4 different transcription factors. So these are proteins that bind to DNA and then change the genes that are being expressed in those cells. And what happens, how you pick for these colonies, is you have a GFP reporter gene that's being driven by an OCT4 promoter. So OCT4 will bind to its promoter and express this GFP protein, and what you can see under the fluorescent microscope is little mounds of cells that are going to be green. So this is actually something that I did this summer. I was able to induce pluripotency. So you can see all these colonies then, and you pick these colonies, and these are the pluripotent stem cells. and then you can do your downstream, I guess, functional assays using those specific cells, which is crazy cool. However, due to safety issues and certain technological restraints, iPSCs are not currently a clinically viable option for regeneration. However, different methodologies and safety concerns are being addressed. So again, it will be interesting, I think, to see where the field is in the next 20 years or so. But something that we did this summer is we were able to show then that OCT4, which is one of the primary reprogramming transcription factors, is in microvesicles of murine embryonic stem cells. So here's CD63, right, which is a microvesicle marker. And we were able to show that OCT4 protein is in these microvesicles. So these stem cells are releasing these factors. And this is in agreement with published reports, which is pretty cool. Actually, all 4 of those transcription factors have been reported to be in stem cell-derived exosomes. But furthermore, microRNAs, so there's this microRNA 290 cluster, which includes these family of microRNAs. So these are highly, highly expressed in embryonic stem cells. And they have all been shown to be in microvesicles that have been isolated from embryonic stem cells and can actually be transferred to fibroblasts or to skin cells. Although how that transfer functionally affects those cells still remains to be seen. But still, I think it's a really interesting concept. And especially interesting with the recent reports, I think in just the last year, 2 years ago, where fibroblasts can be turned into induced pluripotent stem cells using only microRNAs. So this is in the complete absence of any of those transcription factors. So if you introduce certain microRNAs, you can promote iPS cell formation, which I think, again, is really pretty cool. So this kind of leads us to our main goal, and this is more of a gene therapeutic approach to regenerative medicine. But it would be really cool if we could use microvesicles as vectors for promoting in situ repair of damaged tissue. So recently, this group, I can't remember where they're out of, UCSF, I think, in San Francisco, but in a mouse model, they induced heart attack, and then they, in the actual tissue, gave these cells 3 transcription factors, GATA4, MEF2C, and TBX5, and they were actually able to turn cardiac fibroblasts into cardiomyocytes in the actual organs. So there was no petri dish work that needed to be done at all. And these cells that were induced to become cardiomyocytes actually promoted the functional recovery of this, infarct heart. So the size of the infarct decreased, the strength of contraction in the ventricle returned, the ability of these animals to pump blood overall increased. So kind of taking a step aside then, microvesicles can be engineered to target specific tissues. So microvesicles can be engineered to target cancer cells, and they can be targeted to engineered to target only neurons and deliver a molecular payload. So if it's a protein or a prodrug or siRNA, something of that type, that is something that can be done. So it would be really interesting, I think, if we could take these microvesicles and use them as a vector to deliver these 3 transcription factors and target these microvesicles right to the infarct heart. Because that would be a much more efficient way and a much more safe way of introducing these transcription factors than how they did it in this paper here. And that's kind of where we come in, right? I mean, we are the future scientists, and we're going to fund this research, and I think that it's really kind of an exciting avenue for us to explore in a clinical sense. But just kind of with that, I'll be happy to answer any questions, and thank you very much for your time.
Keith, or Rich, did you have anything from Twitter?
Yeah, we're getting some questions in, and relating to especially the last couple of slides you showed about regenerating cardiac muscle, Keith. Are those factors, do they have to be continuously infused to the area, or is it a one-time application of these factors? Do you know?
So I guess I'm not. So I just kind of skimmed the paper. I didn't really tear through it. But I would, well, you know what they say about assuming. So I guess I don't know.
Okay.
But if they're anything like iPS cells, it's going to be a situation where you expose them to the transcription factors, and then that's going to lead to changes in gene expression, which can then persist throughout the lifetime of that cell. So you should only need to introduce it once. But again, and that's just in the context of iPS cells.
Okay.
I have a question for you, Keith. These iPS cells, they are skin-derived precursor cells, correct?
They can be. You can use any differentiated cell, but yeah.
Now, do they have the same factors being released as the mesenchymal stem cells?
So that is one we don't know. iPS cells are still a new enough technology that we're still trying to work out on a mechanistic level, the changes in chromatin dynamics and gene expression, what's occurring. And a lot of research is actually going into trying to increase the efficiency of inducing pluripotency. So right now efficiency is, I think, at 0.002%. So it's a very low efficient process, and I think a lot of people right now are focusing on how to increase efficiency so we can get more cell numbers. But as far as to answer your question, we don't know yet.
Well, I just was concerned with the factors that are currently being released by the MSCs. The ERK1/2 going up, VEGF going up, EGF going up, caspase activity going down,
right,
and being part of a brain tumor lab. These are all things that are bad for us. Right. But I know that a lot of people are engineering these MSCs to carry drugs or viruses or to secrete immune factors that would stimulate a response. But are these other factors being released, from what you're seeing, do you think they might override any positive effect on brain tumor? I mean, these all seem to be pretty negative factors.
Yeah, so there are certain studies, and Ryan probably know more than me, but where mesenchymal stem cells do actually home to tumor sites. And then what they do once they get there, I think, really is context dependent and just how the researchers are looking at it. But there are reports that mesenchymal stem cells can actually kill tumors. And there's actually a girl I was working with who's from Brazil, and her lab works on osteosarcoma, and they used dental pulp-derived stem cells. And in co-culture assays, these stem cells actually killed tumor cells and were inhibitory to tumor formation. And I think they used a mouse model or a dog model. So there's definitely some type of a dichotomy there, I think, that would be interesting to dissect.
Yeah, I know there was a paper, in Nature in 2007 that actually showed that the MSCs found within the tumor stroma actually promoted breast cancer movement, and so there are also plenty of things that show that they're bad as well. So yeah, it's just interesting to figure this out. But
I agree. And it's really kind of interesting too to see the parallels, I think, between a wound healing response and a tumor, because in a tumor, especially in glioblastoma, there's so much necrosis going on, which would be pretty representative, I think, of following tissue injury as well. So some of the molecular pathways involved, I think, it's just some interesting parallels could be drawn.
Hey Keith, I have a couple of questions for you. First, I want to thank you for a very clear, very concise presentation. I enjoyed that very much. So with the mesenchymal stem cells, are they honing to the site of injury for regeneration purposes using the same factors that white blood cells, for example, are honing, or do we know that?
I think it's pretty similar. I mean, it is chemotaxis. As far as if it's the exact, CXCL or CXCR or CXCL12 or SDF1, something like that. I don't know that. Trying to think, sorry. No, so I don't know that it's exactly known, but it is definitely chemotaxis. So it is in response to cytokines and chemokines that are released at the site of injury.
And then are they doing something, because you showed that you can get regeneration. Are they doing something to stop that collagen formation? I mean, that's the big drawback to normal wound healing, is
Right.
that big growth of collagen.
Yep, and I guess I did make a big stink about that at the beginning and then didn't address it later on, so I apologize for that. But yeah, so TGF beta, or transforming growth factor beta, actually inhibits fibrosis in renal regeneration and kidney regeneration, and that is one of the factors that is released by the mesenchymal stem cells. So that does seem to inhibit the amount of collagen or extracellular matrix that's being produced. And then kind of something I didn't touch on, but in cardiac regeneration, people have isolated, I guess, cardiac progenitor cells that release microvesicles that contain the molecule basogen, which at the tumor center we're pretty familiar with that. But it can actually activate different metalloproteinases in heart tissue, and in that way it can degrade some of that extracellular collagen, which would be inhibitory to heart function. So I'm sure there are several different mechanisms in place to inhibit that from occurring. Does that answer your question?
Sure.
Okay.
Very.
We have a question, Keith, from Google Plus from Luke. Luke wants to know, what are some of the safety concerns with iPS cells?
So one of the big safety concerns with iPS cells is going to be whether or not you get tumor formation from occurring. So one of the functional assays that people use to show that they've induced pluripotency is whether or not you can form what's called a teratoma in mice. So what they do is they inject these iPS cells into mice, and then a tumor forms, and then you should get cell types from each of the 3 germ layers, endoderm, ectoderm, and mesoderm, within that tumor. So obviously we don't want that to occur in patients. That would be not helpful. So finding a way to take these iPS cells and then direct them to differentiate into cardiac cells or kidney cells, and then to ensure then that all of the cells in that population are terminally differentiated and no longer pluripotent. I think that's a big safety concern. Also, the most widely used method of inducing pluripotency is using retroviral vectors. So you're introducing this viral vector into these cells to make them pluripotent, so then you can get random insertion of these genes, which could turn on cancer-causing genes in these iPS cells. So again, if you insert or inject that into a patient, that could promote tumor formation as well. So some of those types of safety concerns I think need to be worked out. Also, we need to increase the efficiency of the process. Like I said before, it's about 0.002% efficient. So really, to get enough cells to make this a viable option, we would need to increase that dramatically.
Keith, I have another question for you, and it goes back to the regeneration bud in an earlier slide, and the limbs that were severed and they were able to fully regenerate. What type of cell, at what level do these cells have their potency, or are they pluripotent, multipotent, and what type of cells are you finding in this bud?
Yeah, so this is actually really cool. So like following limb amputation, immediately after limb amputation, all cells within a 100 micron region of the amputation undergo just a generalized de-differentiation. So they go from, so for example, myofibers go from multinucleated to single nucleated. They re-enter cell cycle, and then they proliferate, and then once you reach, I think it's like 10,000 cells, then they start to differentiate again to form, the limb structure. So I guess to answer your question directly, they de-differentiate, and they're multipotent at that point. So de-differentiated muscle cells give rise to muscle cells in the new limb. They will not give rise to new skin cells in the new limb. Does that make sense?
Yeah.
Okay. But yeah, that's a pretty neat phenomenon, I think, that happens in amphibians.
Keith?
Yeah.
I have a question regarding the mesenchymal stem cells and kidney injury.
Mm-hmm.
So I guess the main question is, will our own native mesenchymal stem cells migrate to a site of injury and release these appropriate factors to spur regeneration, or are these just transplanted stem cells that will do that?
So that's a really good question, and I don't know that anyone's definitively shown whether or not endogenous mesenchymal stem cells will migrate to and engraft into the tissue. It stands to reason maybe that these mesenchymal stem cells, I mean, they very much reside within a defined niche within the body, and they might be less inclined to leave that environment. Whereas with these implanted or injected cells, a lot of times, at least in these mouse models, they're injected intravenously, so they're already in systemic circulation. So they're maybe more exposed to some of these chemotactic factors that promote migration to the injured tissue site. So I think just through observations of our general lack of ability to regenerate, our endogenous mesenchymal stem cells are not enough to really get full functional regeneration. So we probably would need the transplantation to kind of kick up our own wound healing response to a sufficient level.
Excellent.
Hey, Keith.
Yeah.
Last week, in our journal club, we discussed your review article, and one of the questions that came up, looking at your first figure, it shows exosomes, and we know that there are certain proteins, microRNAs, etc., that are preferentially accumulated in those exosomes. Do we have any idea the mechanism of that accumulation?
No idea. No idea. So obviously, there are, like you said, microRNA and messenger RNA that's enriched in the exosomes compared to the parents. Recent reports have shown that is it RISC? Yeah, RISC, which is involved with microRNA maturation, is actually associated with endosomes. So that's a physical interaction. So that could be one explanation as to why microRNAs that are in high abundance within a cell could be found in these exosomes. Also, other microRNA processing or RNA binding proteins in general, such as argonaute and some stress-related proteins that bind mRNA, have been shown to be in exosomes and microvesicles as well. So it could be through like a protein-protein RNA kind of interaction that they're enriched and packaged. But in my opinion, I don't think anything conclusive or very convincing has come out to say that this is the pathway that specifically enriches for these molecules. So
that's there again a whole other area that our students could investigate.
Yeah, that's job security is what that is.
Yeah, I'm glad you.
Or a little frustration.
Yeah, right. Exactly. A little bit of both, maybe. Yeah.
I'm glad you brought the discussion back to exosomes, Rob. And another question that came up was, if exosomes are releasing factors that are pro-tumor, Are there mechanisms that we know about that could increase or decrease the amount of exosomes that are being released?
Yeah. So in the context of like tumors, are we talking?
Just any.
Oh.
Is there a way that we can increase exosomes or decrease exosomes?
So I know people have shown that hypoxia, if you're in a hypoxic environment, you release more exosomes. If the cells undergo cell division at a higher rate, they release more exosomes or microvesicles. So if they're more metabolically active. Further, so people are starting to kind of parse out the pathway that leads to multivesicular bodies fusing back with the membrane versus them just being degraded in lysosomes. And some of the RAB proteins, which are small GTPase proteins that are involved with vesicular trafficking, it seems like RAB26 and RAB27 target multivesicular bodies to the cell membrane. So if you could somehow modulate their activity, if you could increase their activity, you would get more exosome release. You know, what that would do to other processes within the cell, I don't think is maybe quite as clear. But yeah, there's this, I can't remember, her name's Thérèse. She's from France. She's in the Marie Curie Institute, and that's really what a lot of her research has been focusing on is, you know, how can we modulate this exosome pathway to increase or decrease microvesicle release.
Thank you.
Yeah.
Do we have any other questions, Rich?
Yep, we just got one from Twitter. Keith, do you know, are there models of, say, stroke, spinal cord injury, Alzheimer's that are being investigated in terms of, you know, tissue regeneration and the role of mesenchymal stem cells?
Yeah, definitely. So I'm not as familiar with the spinal cord injury and mesenchymal stem cells, but I know in a model of ischemic stroke, people were able to, or this group was able, actually they were out of Henry Ford down in Detroit, which was pretty cool. They showed that mesenchymal stem cell derived microvesicles actually promoted neuron regeneration following ischemic stroke. So again, that's in a rat model. But so not as well studied as the kidney field, but there's definitely, I think, applications that are being pursued there.
Thank you.
I am not getting any questions from Facebook, and I'm out of questions, and so unless you gentlemen have more, I'd like to thank Keith for his time.
Yeah, I would too, and I think this is a remarkable field with, you know, just unbelievable application for relief of human illness. And I think, you know, thanks, Keith, for giving us an introduction to this and spurring some discussion, and hopefully some of the younger scientists watching the broadcast today will be inspired to pursue questions in this field. So I think it's really great that you participated with us today.
Yeah, well, thank you very much for having me. I really enjoyed this. Thank you.
All right, well, with that said, I'd like to thank Keith and say goodbye, and maybe Rob has one more thing to say as well, and we'll end this show.
All right, well, thanks. And I also want to thank Keith. Also want to thank Drs. Rovin and Lawrence. There's a fair amount of effort that goes into putting together the logistics and getting people in place and leading some of us neophytes through what it takes to put this together. And so thanks to all those who joined us tonight. Thank you for your participation via Google Plus and Twitter, and we'll do this again soon. And so have a good and safe evening.
And a quick shout out to our sponsor from WeSearch over there. Thanks for helping set this up.
Thank you, guys. It was a little over my head, but I enjoyed it.
Awesome. Well, thanks again, Keith. We'll talk to you soon.
Thank you. Sounds good.
Bye, folks.
Hey, good night, all.
Not yet.